Good morning to all. My name is Agash Maradagam and I am from National College of Pharmacy. Today we are going to discuss about drug metabolism, especially phase 1 and phase 2 metabolic reactions. So we are concising all the metabolism to cover within 10 minutes or 15 minutes. So it's a concised form of lecture on drug metabolism. So drug metabolism is the metabolic breakdown of drugs by living organisms, and our liver is the primary site for metabolism. The liver contains necessary enzymes for metabolism of drugs and other xenobiotics. See the liver. Although the liver is the primary site for metabolism, virtually all tissue cells have some metabolic activities. Other organs having significant metabolic activities include GI tract, kidneys, lungs, etc. Metabolism is often mainly divided into two phases, phase 1 and phase 2 metabolic reactions. There is also phase 3, but phase 3 is more about drug transport rather than metabolism. Some drugs undergo just phase 1 or just phase 2 metabolisms, but more often all the drugs undergo phase 1 first, then it undergoes phase 2 sequentially. Coming to the next slide. Phase 1 reactions of drug metabolisms involves oxidation, reduction or hydrolysis of the parent drug, resulting in its conversion into a more polar molecule. Please note that in phase 1 reactions, the drug metabolism happens and the parent molecule is converted into a more polar molecule. And phase 2. In phase 2 reactions, conjugation by coupling of drug happens. It involves glucouronidation, acylation, sulfation or addition of glycine. The substances that result from the metabolism, phase 2 metabolism may be inactive or they may be similar to those original compound or parent compound. Either they may be similar, either they may be inactive or they may be therapeutic active. So the difference between phase 1 and phase 2, we have to note that. So coming to the first reaction, phase 1 metabolic reactions. See, importantly in this lecture we are going to discuss mainly phase 1 and phase 2 metabolic reactions. First we will go through phase 1 reactions, and here we are going to see some examples of phase 1 metabolic processes, and hopefully this will allow us to predict the outcome of these phase 1 reactions. The phase 1 metabolic reactions include oxidations normally performed by cytochrome P450 enzymes. Oxidations normally performed by cytochrome P450 enzymes. As I mentioned earlier also that it is there are other than oxidation we have reductions and hydrolytic reactions. So let's quickly go through some examples. First we will go through oxidation reactions. I have taken here 3 examples: losartan, carbamazepine and dapsone. First molecule is losartan. Here we can see losartan undergoes oxidation to form, the losartan get oxidized, and the primary alcohol here, losartan has a primary alcohol, and that primary alcohol is oxidized into a carboxylic acid. This is the first example, losartan, only the alcohol is oxidized into carboxylic acid. Let's see the second example of carbamazepine. Carbamazepine. It's a structure of carbamazepine. Carbamazepine, you can see here it's an alkane. Carbamazepine has an alkene here. And we all know that alkenes are the potential sites for oxidation. In this case, in this particular case of carbamazepine, the alkene here is oxidized into an epoxide. See, the alkene is oxidized into an epoxide. So we have seen 2 reactions, of one of losartan and second for carbamazepine. Third, we will go to another example, dapsone. We all know dapsone, that is 4,4-diamino diphenyl sulfone is dapsone. So in dapsone, oxidation oxidation occurs to, we have an amino group here oxidized into an hydroxylaminose product. Here in diapsone, the amino group is oxidized into hydroxylamine as a product. All these reactions, not that I have arranged all the three reactions simultaneously or together in a single slide. All these reactions are mediated by cytokine. First we have seen the oxidation. Now we are going to the second portion, that is dealkylation reactions. Technically, dealkylation is also oxidation. We are losing parts of molecule, the carbon parts of molecule. So dealkylation, or we can say it is oxidation also. The first reaction or the first example starts with codeine. We all know the structure of codeine. There is a methoxy OCH3 group in the codeine, OCH3 group. Notice that this OCH3 group is gone in the product. That is an oxidative process on carbon. As a result, you break this CO bond. See, the CO bond is and the product is formed. So this is another example of dealkylation which comes under the phase 1 metabolism reaction. Another example is fluoxetine. Here N-methyl group is there, NCH3 group. In the product you can see there is no NCH3 group, no N-methyl group. Again, these are oxidations and they are mediated by our cytochrome. The two differences in the differences between oxidation and dealkylation is that both the OCH3, the methyl in the OCH3 in codeine and the methyl in the NCH3 of fluoxetine is gone. So by these reactions, you don't think that phase 1 reactions are all oxidations. We have reductions in phase 1 reactions. So we will see some reductions of phase 1 reaction. See, reduction. Our first drug in the reduction reaction is, I have taken chloramphenicol as an example. We all know the structure of chloramphenicol. Chloramphenicol has a nitro group. Here we know that nitro group when undergoes reduction to form amino group. Here in chloramphenicol, the nitro group in the chloramphenicol is reduced to amino group in the product. This is the only thing happening. All these products are more polar. We have seen oxidation, we have seen dealkylation. All the products are more polar than the parent group. So here we will see another example, second example of reduction, that is nabumetol. Nabumetol contains a ketone and this ketone can be reduced into its corresponding alcohol. So first we have seen chloramphenicol, the nitro group is reduced to amino group. Here nabumetol has a ketone and the ketone can be reduced into corresponding alcohol. See the two products you can see. First one is of chloramphenicol and second one is nabumetol. We can see the amino group in the product and the alcohol in the nabumetol also. These reduction reactions are not typically performed by the cytochrome P450s. It is performed by a whole family of reducing enzymes found in our body. We have a lot of enzymes in our body that can perform these type of reactions. For example, in phase 1 reactions, other than oxidation and reduction, we can say hydrolysis or hydrolytic reactions. The third one. First we have seen oxidation. Second one we have seen dealkylation, but it is also oxidation. We can say that then we have seen reduction. Now we are going to hydrolysis or hydrolytic reactions. The first example shown here is of enalapril. We know we have studied the chapter prodrug and we know much more about enalapril. Enalapril contains an ester and esters can be hydrolyzed in the body to form acids. There are many enzymes in our body which converts esters into acids. Say for example, in plasma we have esterases. There are lipases that break down fats which are esters. So here is the example of enalapril. So these are the main, I have quoted a few examples to get an idea of the phase 1 metabolism. Now we will go to phase 2 metabolic reactions. So phase 2 metabolic reactions. So phase 2 metabolic reactions are often called as conjugations. They often begin with an OH group or an NH group on a molecule, maybe on your drug or perhaps on your another metabolite also. We take that OH group, we take the OH group, we use it as a handle to connect to a more polar group to our molecule to conjugate a polar group to our structure. These phase 2 metabolism reactions are very often occurring on phase 1 metabolites. See, we have seen many phase 1 metabolites. That is, the parent compound is converted into a more polar compound. We have seen that in the phase 1 reactions. That is oxidation, reduction, hydrolysis. We have seen that the parent compound is converted into a more polar group. Mainly, it is not at all necessary that phase 2 metabolism happens only on that metabolism, but very often phase 2 metabolism reactions happens on the phase 1 metabolites. So the phase 1 metabolic reaction will create the OH group, and then that OH group can react in phase 2 reactions. Phase 2 metabolic reactions tend to be very polar and they are readily excreted in the kidneys. So I am just showing two examples for phase 2 reactions. One is sulfation and another is glucouronidation. On the first reaction, so in the first reaction we have a molecule which has an OH group. This OH group serves as a handle to put on a sulfate, and this reaction is sulfation. The OH group serves as a handle to put on a sulfate, and this is sulfation. I will show another example of sulindac. We all know the structure of sulindac. Sulindac here also has an OH group. It's a part of the carboxylic acid, and at that carboxylic acid we introduce the sugar-based carboxylic acid. You can see here the sugar-based carboxylic acid is attaching to sulindac. This is called a glucouronide. And also one other thing happens here is the sulfone. The SO group is oxidized into SO2 group. Actually, this is actually a phase 1 step. And both these reactions occurs on sulindac. Not that both our products here, the sulfate is a very polar group and our glucouronide is a very polar group. So both these reactions happen on sulindac. Phase 2 conjugations also detoxify our metabolites also. Another thing about another important thing about phase 2 metabolism is the detoxification pathway which involves glutathione. This is some structure of glutathione. Glutathione is mainly involved in detoxification. Glutathione is a fairly large molecule. The reactive part is this thiol. This thiol here is the reactive part. The structure of glutathione is normally abbreviated as GSH. Some phase 1 oxidations of drugs can form highly reactive metabolites, and these reactive metabolites can then damage the liver, which is where most of the metabolites form. All our metabolites reaches our liver, and liver has to do the detoxification. So the liver therefore has a relatively high concentration of glutathione. Liver relatively has a high concentration of glutathione. Glutathione reacts with these metabolites in a detoxification process before the metabolites can cause liver damage. Glutathione reacts with the metabolites and it starts a detoxification process before the metabolites. I will show you a classic example of glutathione detoxification, that is by paracetamol. We all know that excess intake of paracetamol is hepatotoxic. Paracetamol undergoes phase 2 oxidation to form a metabolite known as NABQI. NABQI. This NABQI is very reactive and potentially damaging to our liver. The glutathione present in the liver reacts with this NABQI to form a conjugated metabolite that is less reactive. So our liver, I have already told that our liver has a excess storage of this glutathione. Glutathione comes out and it reacts with NABQI, and it forms a conjugate, and which is less reactive. So it is a classic mechanism of detoxification by our liver. So the reaction is here, and our product is on the right. Now this is less reactive. This product is less reactive, and this conjugation is an example of phase 2 metabolic reaction. Furthermore, the glutathione group here helps the metabolites to be excreted by the kidneys. Coming to detoxification, dose size is also important. If a drug has high dose and it forms reactive metabolites, then the glutathione levels can be depleted in the liver, leaving the liver poorly protected from damage. So that here comes the importance of potency. The drug dose can be minimized if the drug potency and efficacy are as high as possible. If a small amount of drug is reaching there, or small amount of NABQI like things are reaching the liver, only small amount of glutathione is required to detoxify that. But when the dose size is higher, it will be all the glutathione levels will be depleted from the liver. So we have seen some examples of phase 2 metabolism and how the phase 2 metabolism can detoxify the metabolites or help to make the metabolites more polar. The metabolites are getting more polar so that the metabolites or the molecules can be excreted by the kidney. So this is a concise form of drug metabolism. Hope you all understand it. Thank you so much. We'll see you in the next class. Bye.